We're working from a really dense, but I have to say fascinating text today. It's a chapter called The Biochemical Roles of Transition Metals by Peter J. Kennelly.
Right.
And I'll admit, when I first saw that title, my brain immediately went to, okay, biology. That's organic matter. When I think of a human being, I think carbon, hydrogen, oxygen. That's the stuff of life, isn't it?
It is. I mean, for the most part. If you were to theoretically analyze the mass of a human body.
The famous blender analogy.
The blender analogy, which, please, nobody do this at home, but if you did, about 97% of the mass would be just 6 elements: oxygen, carbon, hydrogen, nitrogen, sulfur, and phosphorus.
Okay, so that's the organic bulk.
That's the organic bulk. That's the meat and potatoes of existence, as it were.
But that leaves a gap, a 3% gap, and I know a huge chunk of that is calcium, right, for our bones and teeth.
It is, yeah. Calcium takes up most of that remaining 2 or 3%. But if you strip that away, you're left with this tiny, almost negligible fraction of your body's weight.
And it's inorganic.
It's inorganic. It's metal. And that is what we're focused on today, this idea of the inorganic within us.
I love that framing, because it immediately highlights this weird dependence.
It's everything, because without that tiny fraction of a percent of inorganic metal, that organic 97%, it just becomes inert. It stops working. You cease to exist.
So we're talking about things like iron, zinc, copper, manganese, elements I really associate more with, frying pans and electrical wiring and batteries, not my own liver or my bloodstream.
And that's the disconnect we want to bridge. These are the transition metals. If you look at a periodic table, we're talking about that block in the middle, columns 3 through 11.
Right.
Specifically, we're looking at iron, manganese, zinc, cobalt, copper, nickel, molybdenum, and a few others. These are what nutritionists call the trace micronutrients.
And the mission for this deep dive is to really get into the mechanics. We're not just going to say, eat your spinach because it has iron. We want to understand how an individual iron atom actually functions to push an electron around to keep your heart beating.
Yes.
Because there's a paradox here that Kennelly, the author, brings up right at the start, and it really sets the stage.
The central tension of the whole chapter, I'd say, is the central tension of this whole field of biology.
And it's this. These metals are absolutely 100% essential for life. A deficiency in any one of them can cause devastating, horrible diseases. But if you have just a tiny bit too much...
They're incredibly toxic, potentially carcinogenic, cell-destroying poisons.
It's the ultimate biological high-wire act.
It really is. Too little is fatal. Too much is fatal. Your body has to maintain these metals in these incredibly tight, specific ranges. And how it does that, how it cages them, how it moves them, how it utilizes them is honestly one of the most sophisticated feats of engineering in all of nature.
Okay, so let's open up the toolkit, section 1 of our breakdown, the chemical toolkit. Why these metals? Why did evolution reach into the periodic table and say, I need that specific block? Why not just build us entirely out of carbon and nitrogen? What's so special about them?
It's definitely not random. It comes down to 2, let's call them chemical superpowers that transition metals possess, which the main organic elements like carbon just don't have.
Superpower number 1, multivalency.
Multivalency.
Okay, break that down for us. I hear valence, and my mind goes back to high school chemistry class drawing electron shells. But why does multi matter so much here?
Okay, so think about an electrolyte in your sports drink, like sodium, which is Na+, or magnesium, MgO2+.
Right.
In a biological setting, magnesium is always +2. It gives up 2 electrons, and then it just sits there. It's reliable, it's stable, but chemically, it's kind of static.
It's a brick. It builds things, maybe, but it doesn't do things. Is that a fair way to put it?
That's a great way to put it. It's a structural component, but transition metals, they're chemical shapeshifters. They have variable charge states or oxidation states. Iron is the classic example. You can easily flip between the ferrous state, which is Fe2+, and the ferric state, Fe3+.
So it can gain or lose an electron pretty easily.
Very easily. Copper cycles between Co+ and Co2+. Molybdenum is even wilder. It can cycle between +4, +5, and +6. It's incredibly flexible.
And why is that flipping ability useful? Is it just chemical gymnastics?
It's everything. Because it makes them the perfect redox engines.
Redox, that's reduction and oxidation.
Right. And just as a quick refresher for everyone, oxidation is losing an electron. Reduction is gaining one. Oil rig.
Okay.
Because these metals can hold different charges. They can perform this beautiful dance. They can pick up an electron over here, hold on to it, carry it across the cell, and then drop it off over there. They are, in a very real sense, the batteries and the wires of all biological energy.
So when we talk about metabolism or burning food for energy at
Mhm.
the most fundamental chemical level, we're just talking about a controlled process of shuffling electrons from our food molecules over to oxygen.
That's all it is, a bucket brigade of electrons.
And these metals are the buckets in the bucket brigade.
They are the buckets, precisely. Without that ability to shuttle electrons, which we call being redox active, the electron transport chain in your mitochondria would grind to a halt instantly.
And that's where we make ATP, our energy currency.
Exactly. No redox active metals, no APP production. You wouldn't have any energy. You'd be dead before you finish the sentence. It's that critical.
Okay, that's a powerful point. So that's superpower number one. They are the movers and shakers electrons. But the text mentions a second superpower, something called Lewis acid activity. Now, that phrase, that definitely brings me back to some high school chemistry trauma.
We can make it painless, I promise.
Please do.
Mm.
What is a Lewis acid in this context, and how is it different from all the redox stuff we just talked about?
So simply put, a Lewis acid is an electron pair acceptor.
Acceptor.
So think of it less like a bucket carrying electrons and more like a magnet for electrons.
Okay, I like that. A magnet.
Some metals, and the key players here are zinc Zen 2+, and to some extent manganese, are often not used to move electrons back and forth like iron is. They're not doing the bucket brigade. Instead, they sit in the center of an enzyme and they tug on the electrons of the molecules around them.
You create a kind of chemical tension.
That's a fantastic word for it. They create tension. Let's take a water molecule, H2O. It's pretty stable. But a zinc ion in an enzyme can grab onto that water molecule and start pulling on its electrons. This makes the oxygen atom in the water very agitated. It makes it more nucleophilic, which is just a fancy way of saying it's more reactive and ready to attack something.
So the zinc turns a mild-mannered water molecule into a chemical saw.
Yes, exactly. And this is absolutely critical for what we call hydrolytic enzymes.
Hydrolytic. Hydro for water, a lytic for splitting.
Enzymes that use water to break chemical bonds. If you want to digest a protein from your lunch, you need to cut the bonds holding it together. Your enzymes use water to do that, and zinc makes the water sharp enough to make the cut.
Okay, let me try to recap this to make sure I've got the dichotomy right in my head. We have one team of metals, the redox engines, like iron and copper, that are all about moving energy around by passing single
The
electrons.
bucket brigade, yep.
And then we have the other team, the Lewis acids, like zinc, that act as static chemical activators. They don't move, they just pull on electron pairs to make other molecules like water more aggressive so they can break stuff down.
That is the fundamental dichotomy of the transition metals in biology. You've nailed it.
But this is where it gets tricky. Section 2 of our outline is the safety suit. Because you mentioned earlier how toxic these things are, and the text uses a very specific, very evocative phrase, naked ions.
Right, and you basically never ever find a naked or free-floating iron or copper ion in a healthy cell. It would be a catastrophe.
Why? What would it do?
Well, they're chemically promiscuous.
Promiscuous.
They will react with anything and everything. If you just had a naked iron ion floating around in your cytoplasm, it might start ripping electrons from your DNA or from the lipids in your cell membranes. That's damage. That's toxicity.
Okay, so that's the reactivity problem.
And there's a solubility problem. Simple rocks don't dissolve well in water.
Yeah.
If you just had iron filings in your blood, they'd just clump up and sink. They wouldn't be useful.
So the body has to solve both problems at once. It has to put them in a cage.
That's the perfect analogy.
Yeah.
This process is called complexation. We take the metal ion and we incorporate it into what's called an organometallic complex. Basically, we build a cage of organic molecules around it.
And the bars of the cage are proteins. The text mentions side chains like aspartate, glutamate, histidine, cysteine. These are all amino acids.
Those are the bars of the cage, yes.
Yeah.
Those specific amino acids have atoms in their side chains, like the sulfur and cysteine or the nitrogen and histidine, that have lone pairs of electrons that can reach out and form a coordinate bond, chemically gripping the metal.
So they grab onto it.
They grab it and hold it tight. Sometimes, for really important metals like iron and hemoglobin, we use an even bigger, more complex organic ring structure, like a porphyrin ring, to hold the metal right in the center.
And this complexation, this caging, it solves the solubility problem.
It solves 4 problems, actually. First, yes, solubility. Wrapping it in a protein makes the whole complex soluble, so it can float around in your blood or inside a cell.
Second, reactivity control. It stops the metal from burning down the house, from reacting with things it shouldn't. Third, it helps assemble multi-metal units, which we'll get to later when we talk about teamwork. But the fourth benefit is, for me, the coolest one. It's called tuning.
The fine-tuning concept. I loved this part of the chapter because it implies that protein isn't just a passive holder, a cage. It actually changes the personality of the metal inside.
It absolutely does. It changes what we call its standard reduction potential. But let's use a better analogy. Imagine you have a generic engine, a really good engine.
Yeah.
If you put that engine in a speedboat, it's tuned to do one specific thing. High RPMs, push a propeller. If you put that same engine in a generator, it does another thing. Constant, steady output. If you put it in a race car, it does a third thing. The iron atom is the engine. The protein wrapped around it is the chassis.
So an iron atom in hemoglobin is the same as an iron atom in a cytochrome.
Identical. But in hemoglobin, the protein chassis has tuned it to be perfect for grabbing and holding an oxygen molecule. Whereas in a cytochrome, the chassis has tuned it to be perfect for grabbing and then immediately letting go of an electron.
Exactly. And an iron atom in a different enzyme might be tuned to rip apart a specific chemical bond. It's the same iron atom. But the cage, the specific amino acids that are grabbing it, the geometry of how they're squeezing it, it pushes and pulls on the metal's electron cloud to make it perfect for that one specific job. It modulates how easily that iron gives up or takes on an electron.
There's a great visual in the text, figure 10.2, describing something called a zinc finger. I think this illustrates the whole cage idea really well because it sounds almost mechanical.
The zinc finger is an iconic example of this. It's beautiful. Imagine a short loop of a protein chain. On its own, it's floppy. It has no defined three-dimensional structure. It's like a piece of wet spaghetti.
Okay, useless.
Totally useless. But then you stick a single zinc ion in the middle, and 4 amino acids in that loop, usually 2 cysteines and 2 histidines, reach in and grab onto it.
Like 4 fingers gripping a baseball.
That's a perfect image. And that zinc ion acts as a structural pin, a linchpin. It locks that floppy loop into a rigid, stable shape, a finger. And that rigid shape is exactly what's needed to slot perfectly into the grooves of your DNA, double helix, to turn specific genes on or off.
That is just wild to think about. So a tiny, simple piece of metal is literally holding our complex organic genetic instructions open so they can be read. It's like a paperweight for the code of life.
It's precisely that. Without the zinc, the protein goes floppy, it falls out of the DNA groove, and you can't read your genetic code. The whole system fails.
Amazing. Okay, so with that foundation laid, let's get into the specific characters. Section 3, iron. The text calls it the versatile heavyweight.
And for good reason. Iron is the king. It's the most abundant transition metal in the body, and it's because it can do almost anything.
We usually hear it divided into heme and non-heme iron. Heme is the famous one, the stuff in blood, right?
Right. Heme iron is iron that's bound in that porphyrin ring I mentioned, that big, flat, organic donut with the iron atom sitting in the hole in the middle. It's what makes blood red.
And its main job is oxygen transport?
In hemoglobin and myoglobin, yes, its job is to bind to oxygen. But here's that tuning concept again. In a different class of proteins called cytochromes, which are also heme proteins, the iron doesn't hold oxygen at all. It just cycles relentlessly between Fe2+ and Fe3+ to pass electrons down the assembly line. Same heme, same iron, different chassis, different job.
But the text goes really deep into non-heme iron, which I feel like gets no press. We always hear about heme. What is this non-heme stuff doing for us?
It's the real unsung hero of the iron world. The most fundamental type are these things called iron-sulfur clusters, or Fe-S clusters.
Okay.
These are literally just little cubes or clusters of iron atoms bonded directly to sulfur atoms, all tucked inside a protein. These structures are chemically ancient. They are likely some of the very first biological catalysts to ever exist on Earth, maybe even before life as we know it.
Wow. And what do they do now?
They're crucial. They're found in enzymes that are responsible for DNA replication and DNA repair. So the very act of copying your genetic code depends on these ancient iron-sulfur cubes.
And then the text mentions the diiron center, so 2 iron atoms working together.
Yes, sometimes one atom just isn't enough chemical power for the job. In the enzyme ribonucleotide reductase, which is essential for making the building blocks of DNA, you have a diiron center. The 2 iron atoms work in concert to do some really tough chemistry.
Where else do we see that?
You also see it in an enzyme called methane monoxygenase, which some bacteria use to turn methane gas into methanol. It's a very difficult reaction and takes 2 irons to pull it off.
And P450 enzymes. I've definitely heard about these in the context of drug interactions. Don't eat grapefruit with this medication because it messes with your P450s. What is iron doing there?
The P450s, or cytochrome P450s, are the body's primary detox squad. They're mostly in your liver. They use heme iron to oxidize drugs, toxins, pollutants, what we call xenobiotics, to make them more water-soluble so you can excrete them in your urine.
So the iron is basically incinerating the toxin chemically.
It's performing a controlled chemical burn on the toxin. It's heavy-duty redox chemistry, and it's happening constantly to protect you from what you eat and breathe.
And just to show off, the text mentions that iron can sometimes act like zinc. It can play for the other team.
It can. It's rare. But there are enzymes called purple acid phosphatases. They use iron and zinc, or sometimes iron and manganese, and their only job is to act as Lewis acids, to use water to hydrolyze bonds with no electron shuttling involved. Iron can basically do it all. It's the Swiss Army knife of the periodic table.
So if iron is the chaotic, versatile multitasker, Section 4 brings us to the steady hand, zinc, the structural architect.
Zinc is chemically unique among these transition metals we're talking about because it has what's called a full D shell of electrons. Now, what that means for us, in simple terms, is that it cannot do redox. It is always Zn2+. It will not accept another electron. It will not give one up. Its multivalency is, well, it's not multi. It's monovalent in practice.
Which makes it safer, right? Because earlier you said the redox active metals can cause damage.
Much, much safer.
Yeah.
Iron and copper, precisely because they're so good at moving electrons, can sometimes drop one. They can spark. That spark can create a free radical or reactive oxygen species, or ROS, which is incredibly damaging to cells. Zinc doesn't spark. It's stable. It's quiet.
So its two main roles are structural and catalytic. We already talked about the amazing structural role in zinc fingers, literally holding the proteins that read our DNA together. What about catalysis?
This is its big Lewis acid role again. And the classic example, maybe the most important one in the entire body, is an enzyme called carbonic anhydrase.
Carbonic anhydrase. I know this handles cockroaches from breathing, but can you walk me through the mechanism? How does zinc help?
Sure. It's absolutely vital. Your cells are constantly producing carbon dioxide as a waste product from metabolism. You need to get that CO from your tissues to your lungs to be exhaled.
Okay.
But CoGray is a gas, and it doesn't travel very well in your watery blood on its own. Carbonic anhydrase has a zinc ion at its core. That zinc grabs a water molecule, activates it, makes it angry, like we said, and that angry water molecule attacks the CO and instantly converts it into bicarbonate, which is an ion.
And bicarbonate travels well in blood.
Bicarbonate travels perfectly in blood. It dissolves easily. When it gets to your lungs, the enzyme just runs the reaction in reverse, turning the bicarbonate back into CoGray, and you breathe it out.
And this happens fast.
We're talking about one of the fastest enzymes known in nature. It can process up to 1 million reactions per second. Without the zinc to catalyze it, that reaction is far too slow, and you'd effectively suffocate on your own metabolic waste.
Wow. And the text says there are over 3,000 zinc-containing proteins in the human body. That number seems huge.
It's a massive number. It really underscores its importance. It's involved in digestion through proteases that break down protein. It's in enzymes for DNA repair. And as we said, thousands of transcription factors. The zinc fingers.
And scientists are trying to weaponize that in a good way.
They are. People are trying to engineer artificial zinc fingers for gene therapy. The idea is you could design a protein that uses zinc fingers to recognize and bind to a very specific sequence of DNA, say, a faulty gene, and then deliver a tool to edit it. Zinc is truly the bedrock of biological infrastructure.
Okay, moving on to Section 5, copper and manganese. The text groups them as energy and protection. Let's start with copper. I still think of wires, and you said earlier that's a good
It's a
announcement.
perfect analogy. Copper is an excellent electron carrier, a biological wire, and it's most famous for its role in the absolute final step of your respiration. There's a massive enzyme complex called cytochrome C oxidase. It uses 2 copper atoms and 2 iron atoms. Its job is to take all the electrons that have come down the bucket brigade from your food and dump them onto an oxygen molecule to create water. That is the moment you actually breathe at a cellular level. It's the end of the line.
But copper isn't just for breathing, is it? The text lists some other kind of surprising roles. It's in our skin, in collagen.
That's right. An enzyme called lysyl oxidase is copper-dependent. Its job is to form crosslinks between collagen and elastin fibers.
Crosslinks, what does that mean?
Imagine knitting a sweater. The collagen fibers are the strands of yarn. Lysyl oxidase is the needle that comes in and ties strong knots between the strands. It's what gives your skin its elasticity, and your blood vessels and other connective tissues, their strength and integrity.
So without enough copper.
Your tissues would literally be too weak to hold together properly.
And pigment, like hair and skin color.
That's another copper enzyme, tyrosinase. It uses copper to synthesize melanin. A genetic defect in that enzyme, and you can't make melanin, which results in albinism.
And adrenaline.
Dopamine beta hydroxylase, a copper enzyme that converts dopamine into norepinephrine, which is then converted into epinephrine or adrenaline. So your entire fight-or-flight response is copper-dependent.
Wow. Okay, let's talk about manganese. I have to admit, I usually forget about manganese. I confuse it with magnesium all the time.
Most people do, but your mitochondria absolutely love manganese. It is found predominantly inside the mitochondria, the power plants of the cell. It's a key cofactor in the TCA cycle, the Krebs cycle, which is central to energy production. It also helps in the urea cycle to get rid of toxic ammonia.
And it has a special protective role, right? The text mentions MNSOD.
Yes, MNSOD, which stands for a manganese superoxide dismutase. It's a mitochondrial bodyguard.
A bodyguard against what?
So mitochondria are burning oxygen to make energy, but that process, it's messy. It's like a faulty engine that starts a lot. It creates a toxic byproduct, a free radical called superoxide. MNSOD sits right there in the mitochondria, and its only job is to find and neutralize those superoxide sparks before they can set fire to the machinery and cause massive oxidative damage.
It's the bomb squad in the power plant.
That is a perfect description. If you didn't have MNSOD, your cellular power plants would essentially burn themselves down from the inside.
Okay, section 6, the supporting cast. These are the metals we need in really, really tiny amounts. Cobalt, molybdenum, nickel, vanadium, chromium. I feel like these often get glossed over in nutrition, so let's give them their due. Let's do a rapid fire, but with detail. Cobalt.
Cobalt has exactly one job in the human body. One, it sits at the absolute core of vitamin B12, which is also known as cobalamin.
That's it, just B12. It has no other function.
As far as we know, just B12. But B12 is incredibly critical. The cobalt sits in a special ring called a core-in ring, which looks a lot like the heme ring in iron, and it performs a very, very rare chemical trick. It forms a direct carbon-cobalt bond.
And why is a carbon-cobalt bond so special?
It's extremely rare in all of biology, and allows the enzymes that use B12 to move around methyl groups, single carbon units. This is absolutely essential for things like DNA synthesis and for converting a toxic amino acid called homocysteine into the useful one, methionine.
So if you don't have cobalt,
you don't have functional B12, your red blood cells fail to divide properly, and you get a condition called pernicious anemia.
Okay, molybdenum. This one is fun to say.
Molybdenum. In humans, molybdenum is what we call the universal cofactor. It never works alone. It is always bound to this weird, specific organic molecule called molybdopterin.
Molybdopterin, it sounds like a dinosaur.
It's a phaedorin ring, a specific organic structure. And this molybdenum-molybdopterin complex, it powers a few crucial enzymes.
Right.
One is anthene oxidase, which is the final step in breaking down purines into uric acid. Another is sulfite oxidase.
And sulfite oxidase does what?
It detoxifies sulfites, which you get from certain foods, preservatives, and also from the breakdown of sulfur-containing amino acids in your own body.
And the text mentions that if you have a genetic defect in this,
it's absolutely tragic, a condition called sulfite oxidase deficiency. The sulfites build up to toxic levels and cause severe, irreversible brain damage and usually early death. It just shows how critical even these incredibly obscure metals are. One missing piece and the whole system collapses.
Nickel. What's the story with nickel?
Nickel is the mystery. We know for a fact that plants and bacteria absolutely need nickel for an enzyme called urease, which breaks down urea. We also know that humans need trace amounts of nickel. There have been animal studies where if you deprive a goat of all nickel, it fails to thrive. But we have not yet actually identified a specific human enzyme that contains nickel.
So it's a ghost metal. We know it's doing something important. We just can't find where.
Exactly. The search is on.
Vanadium.
Vanadium is interesting because in pharmacological doses, it's known to mimic insulin. It seems to work by inhibiting certain enzymes called phosphatases, which enhances insulin signaling. But again, its essentiality in the tiny amounts you'd get from diet is cryptic. We haven't found a required vanadium enzyme in humans.
And finally, chromium. This one I see in supplements all the time. Chromium picolinate for weight loss and blood sugar.
Ah, the controversy. Back in the 1950s, research identified something they called a glucose tolerance factor. And they thought chromium was its active component.
Okay, so that's where the idea comes from.
That's the origin. But decades of follow-up research have completely failed to isolate or identify a specific chromium-containing biomolecule that does this. The current scientific consensus is that the evidence for chromium supplements is pretty weak, and we aren't even 100% sure it's an essential nutrient in the classic sense anymore.
So if you're buying chromium supplements...
You might just be making expensive urine. The jury is very much still out on that one.
Fascinating. Let's move to Section 7, multi-metal units. We're talking teamwork here, we've touched on this, but sometimes one metal atom isn't enough. Why does nature double or triple up?
Because some chemical jobs are just too tough for one atom. Biology often pairs them up to do the really heavy lifting. Let's go back to urease, that plant enzyme I mentioned that uses nickel. It actually uses two nickel atoms sitting side by side. One nickel atom polarizes the urea molecule, making it vulnerable. The other nickel atom activates a water molecule, making it ready to attack, and then bam, they work together to break the bond. Neither one can do it alone. It's like using two hands to snap a thick stick.
I see. And nitrogenase, that sounds important.
This is the enzyme in certain bacteria that keeps all of us alive. It takes inert nitrogen gas from the atmosphere in and turns it into ammonia, which is the source of nitrogen for all plants and therefore all animals.
So fertilizer, basically.
Natural fertilizer. And breaking the triple bond in years is one of the most energetically difficult reactions in chemistry. To pull it off, nitrogenase uses this massive, mind-bogglingly complex cluster of iron and molybdenum atoms. It's one of the most sophisticated metal centers in all of nature.
And our friend cytochrome oxidase again, the FeCO center.
FeCO center. Iron and copper working together, they have this incredibly delicate job. They have to hold on to an oxygen molecule, O euros, and feed it 4 electrons one by one without ever letting go of it prematurely.
And if they did let go too early...
You get superoxide or peroxide, those toxic radicals we talked about. They have to hold that oxygen tight until it is fully and safely converted into 2 molecules of water. It requires perfect electronic and spatial coordination between the 2 different types of metals.
Which brings us to the scary part, Section 8. The dark side, toxicity. We said it at the beginning, essential but deadly. What actually happens when we have too much or when we get exposed to the wrong kind of metal?
Right. So first, let's just quickly define a heavy metal. Usually in a toxicological sense, it just means a metal with a density greater than about 5 grams per cubic centimeter. So we're talking about the classic villains, lead, mercury, cadmium, arsenic. But it's important to remember that even the essential ones like iron and copper are heavy and become toxic in excess.
And mechanism number 1 for their toxicity is displacement. How does that work?
This is a classic case of molecular mistaken identity.
Sure.
Let's take lead, PbO. Chemically, it looks a lot like zinc, ZnO. They have the same charge and a similar size. If you get lead poisoning, the lead atoms can actually go into your enzymes and kick the zinc atoms out.
Like a fake key jamming a lock.
It's exactly like that. The lead goes into the zinc finger or into the active site of a zinc enzyme. It fits, but it doesn't work. It's a dud. It can't do the required chemistry, so the enzyme just shuts down.
And the text says this is why lead poisoning causes anemia.
Yes. Lead displaces zinc in several of the key enzymes that synthesize heme for your hemoglobin. So you literally can't make new blood because the lead kicked the zinc out of the factory.
That is genuinely terrifying. Okay, mechanism 2. Enzyme inactivation via another route.
Heavy metals, particularly things like mercury and arsenic, absolutely love sulfur. They have a very high chemical affinity for sulfhydryl groups, which are found on the amino acid cysteine. Many proteins rely on these sulfur groups to maintain their specific 3D shape, or they have a critical cysteine right in their active site. When arsenic comes along and binds irreversibly to those sulfurs, it warps the protein. It twists it out of its functional shape.
And structure is function in biology. If you warp the shape, the machine breaks.
The machine is broken. Arsenic poisoning, for example, famously affects the pyruvate dehydrogenase complex, which is critical for energy metabolism, by binding to the sulfurs on a cofactor called lipoic acid. It basically just puts a clamp on your whole metabolic engine.
And the third mechanism, this one sounds like an action movie title, the Fenton reaction.
It's the most explosive one, chemically speaking. If you have excess naked iron, specifically FeO or copper, floating around where it shouldn't be, it can react with hydrogen peroxide, which is a normal low-level byproduct of cell metabolism.
Okay.
This reaction, the Fenton reaction, creates the hydroxyl radical. That's OH with a dot, OH.
And why is the hydroxyl radical so bad?
It is without question the most reactive, indiscriminate, destructive chemical species found in biology. It's that molecular buzzsaw we talked about. It will react with and destroy the first thing it bumps into.
Like what?
It'll rip a hydrogen atom off your DNA, causing a mutation that could lead to cancer. It'll attack the polyunsaturated fatty acids in your cell membranes, causing a chain reaction that rips the cell wall apart. It is pure, uncontrolled chemical violence.
So having excess free iron literally rusts you from the inside out.
That's a very good way to think about it. That oxidative damage is now thought to be a major driver of aging and many chronic diseases.
Which explains then why the body guards the door so tightly. This brings us to the final section, section 9, gatekeeping, absorption and transport.
Exactly. Since we can't really excrete most metals very well, we don't have, say, a specific pump to just pump excess iron out of the body once it's in. Our entire strategy has to be based on strictly controlling how much gets in the first place.
The bouncer at the club door is more important than the guy who kicks people out.
Much more important. And the main bouncer is a protein called DMT1, the divalent metal transporter 1. It sits on the surface of your intestinal cells in the duodenum.
And it lets in.
It's not very specific. It lets in iron, Fio, manganese, nickel, copper, Qa. It basically lets in a range of metals with a +2 charge.
But there's a problem with that lack of specificity, isn't there? A competition problem.
A huge problem. They all have to use the same door to get into the club. So if you, for example, take a massive zinc supplement, the zinc ions will flood the entrance and hog all the DMT1 transporters.
And the copper can't get in.
The copper can't get in?
Yeah.
You can actually induce a severe copper deficiency, which can cause anemia and serious nerve damage just by taking way too much zinc over a long period. They compete for absorption.
That is a crucial tip for anyone listening who takes supplements. More isn't always better. Okay, so once a metal gets inside, how do we move it around safely?
We have specific Uber drivers for them. For iron, the main transport protein in the blood is called transferrin. It binds iron very tightly so it can't cause any Fenton reactions. Inside the cell, excess iron is stored in a giant hollow protein safe called ferritin.
And who's in charge of all this? The text mentions a master regulator hormone, hepcidin.
Hepcidin is the master regulator. It's a hormone produced by the liver. If your body's iron levels get too high, or interestingly, if you have a serious infection or inflammation, the liver releases hepcidin into the blood.
And what does hepcidin do?
Hepcidin travels to the gut, and it tells the bouncer, DMT1, to shut the door. It locks it down. It completely blocks any more iron absorption. It's the body's emergency brake.
Why does it do that during an infection? That seems odd.
Because invading bacteria need iron to grow and multiply. It's an essential nutrient for them, too. So if you have an infection, your body goes into a defensive crouch. It tries to starve the bacteria by hiding all the available iron. That's why you often get anemia of chronic disease when you're sick. Your body is deliberately hiding the iron to protect you.
That is a brilliant evolutionary strategy. Just hide the food from the invaders. And finally, when this whole transport system goes wrong genetically, we get specific diseases, Menkes disease.
Menkes disease is a tragic genetic failure of a protein needed to absorb and transport copper. Children born with it have brittle, kinky hair because the collagen and keratin isn't cross-linked properly by lysyl oxidase, and severe progressive neurological degeneration because their brain can't get the copper it needs for its enzymes.
And pernicious anemia, we mentioned earlier, a failure to absorb B12 and therefore cobalt.
Right. It all just highlights how incredibly fragile and precise this entire system is. You need these metals, but you need them in exactly the right place at the right time, bound to exactly the right protein cage.
So let's try to wrap this all up. We've gone from the periodic table to the mitochondria to the hospital ward. What is the single biggest takeaway for you from this deep dive?
For me, it's the realization of how we are a hybrid technology. We think of ourselves as soft, organic, biological fluff, but we're not. We are deeply integrated with the inorganic geology of the Earth. We've harnessed the chemistry of rocks, of iron, copper, zinc to power, that is most delicate, sophisticated machinery of life.
It really changes how I see a simple multivitamin pill. It's not just abstract health points anymore. It's a supply of raw materials for billions of microscopic engines.
It is. And I want to leave the listener with this one final thought to really bring it home. Think about the zinc finger again.
Okay.
You have this tiny single atom of zinc, an inorganic piece of metal, sitting in the center of a loop of protein. And that metal ion is the only thing physically holding that protein in the correct shape required to read your DNA.
Without the metal.
Without that single atom of metal, the protein collapses into a useless floppy string. The DNA cannot be read. Gene expression stops. Life stops. At the very center of your being, at the interface with your genetic code, You are being held together by a piece of rock.
We are inorganic anchors in an organic world.
We really are.
Thank you for listening to The Deep Dive. Respect your micronutrients, but as we've learned, don't overdo it. We'll see you next time.
Stay curious.